It takes decades for a promising candidate to be developed for use in clinical practice. Unfortunately, with rare diseases such as DMD, we always knew there would be tougher challenges to overcome. For our first session, we invited leading DMD clinicians to discuss therapies used to treat patients today, approaches to early diagnosis and treatment, and the future of newborn screening. Our moderator is Håkan Miller, who is a DMD patient, as well as a featured writer for BioNews, where he tells stories about people making a difference in the rare disease community. As a DMD patient, he also has firsthand insight into living with this devastating disease.
Hello, everyone. Welcome to the WuXi AppTec Charting the Path for New Therapeutics and Better Care clinical panel. And I'm happy to welcome Dr. Richard Parra, Dr. Stanley Nelson, Dr. Kevin Flanagan. Thank you guys so much for your time, and I'm looking forward to the conversation.
Thank you.
All right, perfect. So I'm going to, just jump right into it. This is for you, Dr. Nelson. Right now, there are 5 FDA-approved drugs for Duchenne Muscular Dystrophy. And so why don't you tell me a little bit about the impact of having that many drug approvals in the process by the FDA at this point?
Sure. It's been a seminal shift in the Duchenne field to actually have any FDA-approved drugs. And it's only been to late 2016 the very first drug was approved. So all 5 of these have occurred in reasonably recent history. And it's a steroid. It's 4 mutation-specific exon skipping drugs, and we now have these in our armamentarium for treating Duchenne. Not all patients with Duchenne, but a sizable fraction of those patients with Duchenne. And it's already begging the questions about how do we combine these? When do we start? When should we start an exon skipping drug? And many of us would feel that should be as young as possible. There's an open discussion of when and how young to start steroid therapy. And many of us feel younger, the better, since the disease process is already present at birth. There's no reason not to start trying to mute it as best we can. So, to me, what this indicates is we're heading towards a world where we will be having a multitude of different therapeutics, both mutation-specific and mutation-independent, and we will be figuring out how to use these things in combination to slow the disease and hopefully halt the disease and, in an ideal world, reverse the disease for many of those that are far progressed along the disease course.
Yeah, that's really interesting. Dr. Flanagan, Dr. Parra, do you have any other things to add, about that, when we should start diagnosing and I guess treating the patients with these drugs, as far as timing? Anything else you wanted to add?
Well, newborns are a particularly vulnerable group that make both the FDA and I think many physicians a little nervous because newborns don't necessarily act the way older children do in response to a drug, and children don't necessarily respond the same way as adults. So it gets to be very hard to move therapies that get developed and tested in clinical trials in adults and older children down into the newborn group. So I think there's a challenge there, and it would of course be helpful to be able to detect early groups so that they could be studied more carefully to look at safety and best combinations or individual treatments. But how to do that is a little unclear. I know we'll get into newborn screening a little more, but I think Dr. Nelson and Flanagan have some challenges to moving these therapies down into the very young ages, where clearly I totally agree that starting these therapies before injury is done would be the smartest thing to do.
I'll just add to that, if I could. I think there's really growing evidence that even small amounts of dystrophin expression do change the trajectory of disease over many years. So that's coming out of the treatment trials, but there's increasingly amounts of data from this from patient observations as well, unusual mutations that allow small amounts of dystrophin. So although I think many of us wouldn't necessarily call the 5 approved drugs kind of the home runs we're after yet, they do make a significant difference over time. The data clearly shows that. And I think I agree with Stan's comment that I'm one of those people who agree with very early treatment with those therapies. And I think the 5 things in the past 5 years tells us there is progress. There are other things in the pipeline that are promising, including other chemistries for exon skipping approaches and other exons being targeted. So the field is clearly moving, and we have to think carefully about how these issues of timing and the issues of newborn screening to optimize therapeutic onset.
Yeah, I would say, Hawken, just for practical ramifications, we do occasionally diagnose patients even without newborn screening around 1 to 1.5 years of age. And we have started exon skipping drugs at that relatively young age group and have started particularly weekend only, not necessarily daily dosing of steroids, and particularly the specifically approved steroid for Duchenne in that young group. And we don't have decades of experience with this yet, though. So, but I think we're all reasonably pleased, but we may be pleased based on imperfect data at this point.
Right. It sounds like, to build data, you need time, right? And you have to, be a little bit patient in some cases with that. And I think all of you, including you, Dr. Nelson, mentioned, it's going to take a combination therapy. So, you have the steroid approved, exon skipping drugs approved. What would you guys like to see come out next that could really have a significant impact in addition to what's already approved?
Well, I'll jump in to say I think the most promising at the moment is really gene therapies that'll deliver microdystrophin gene therapies that are in their best implementation will be mutation agnostic, are not dependent on the specific mutation. And we're fortunate now that there are well-designed trials from multiple different companies using slightly different versions of this, where we should get answers in the relatively near term about the benefit of this. That still leaves us, I think, with some challenges in that those are engineered versions of the protein. They're not, we don't have Becker muscular dystrophy patients, let's say, who are walking around with those exact proteins being expressed. We all expect benefit from them, and all the ones in trials show benefit in preclinical studies. We do have to see that data, though, to get a sense of how that is. But really, as an interim step, what we're all after is, of course, full-length dystrophin re-expression therapies. And there are some promising approaches on the horizon for that as well. But clearly, the near term is microdystrophin gene therapies is the most exciting approach.
And for right now, Hawken, they're driven by specific gene promoters. The AAV vectors that are delivering the genes are not getting to every cell that is missing dystrophin. So it's targeting a portion of the cells, but the major cell that's affected, mature myofibers are most dependent on a dystrophin being present. Do we need dystrophin? Would we benefit from dystrophin in satellite cells and the stem cells of muscles? Probably, and that would be a good, relevant sort of next generation. But I agree completely with Kevin that I think the disease is caused, it's a Mendelian disease that's caused by a lack of dystrophin from before birth, and replacing that in the key cells that are needed would be certainly a major step forward. It's probably not, I think we ought not be thinking of that as a cure. We ought to be thinking of that as a treatment, and because we're not going to be curing it, we're not going to be treating it in the cells from the very first step that it was occurring. We're always going to be treating it after the disease is already established. So I think we've got a lot of work to do. The part that I'd be excited about as well with that is recognizing that it's plausible to do it, that we can have AAV treatments, at least, if not other delivery vehicles, and replace with microdystrophin. What else do we need to do to change the muscle microenvironment? Is there a way to alter this? Muscle is very plastic. It absolutely changes over time. It changes during our lives. We replace our muscles, replace our satellite cells. Fibrosis can be reversed in theory. Immune response can be reversed in theory. What's that mixture of treatments that might be borrowed from the cancer field, from other fields that we might be using in the context of gene therapy to make gene therapy even more effective? I think there's a lot that we'll be learning in this next decade.
Yeah, sounds very excited. I mean, there's so much that we will learn and that we can bring from other, disease communities. I think it's very interesting. So I'm going to move into newborn screening. We were talking about this a little bit earlier. I know this is Dr. Prad's specialty, but I wanted to ask you, what is the biggest factor that impacts successful clinical outcomes in Duchenne patients, and how does newborn screening factor into that?
Sure. I should qualify myself at the beginning as not a Duchenne clinician. I'm a neonatologist and a pediatric pulmonologist with mostly experience with cystic fibrosis, but there's been a very, I think, parallel story between Duchenne and CF. And speaking from my experience with cystic fibrosis, it was clear that the earlier the available therapies could be started on children, the better their outcomes were. I think that sort of makes common sense. May not be true for all diseases, but I think there's some evidence in Duchenne that would also be true. So how do you find children who are affected before, as early as possible, and before they have symptoms that may be irreversible? And newborn screening is certainly a principle that's been around for about 50 years and has been very successful from a public health standpoint in identifying many very rare disorders. There's sort of a set of rules that go along with it, which are you may put a disease into a newborn screening panel if there's a way to identify it fairly easily, that a laboratory could do the testing, and that there's a therapy available, which if started early would impact the outcome. And I think Duchenne now, as Dr. Nelson mentioned, since at least 5 years ago, would clearly fit into that category, because we know there's a test that can be easily done, and that's to measure CK levels in the bloodstream as at least a first tier test, and take those babies who have high levels to a second tier of testing that would be genetic testing, either probably sequencing of DMD that could be done on a population basis. And there have already been, I think, over 2 million children worldwide in various small trials that have shown that can be done successfully. So if we can identify children before they have symptoms, and we have therapies available that we can start very early, I think newborn screening is really where all the focus goes once the therapies are available. There are a group of patients already out there who have already escaped, and they're going to still have to be picked up as early as possible, trying to avoid the diagnostic odysseys that I know a lot of Duchenne families go through. But ultimately, if there is universal screening, and that would mean each of the 50 states in the United States, and each country makes its own decisions about whether or not to add a disease to the panel. If that can become fairly universal, so it's fair to everyone to be screened early, then you have a fairly healthy, normal-looking baby that is ready to receive a preventive therapy in a sense, maybe not a cure quite yet, but and then it also does help to have probably registries of to somehow keep track of these babies with cystic fibrosis. There's the advantage of having care centers and a nationwide database so that there can be sort of tracking of newly diagnosed patients and what things are being tried for them and what works and what doesn't. And I think that may also help speed up trying to identify the right combination of therapies to best improve outcome. Because if every center is isolated and sort of has their own experience and it's not being shared or can't be analyzed in a scientific way, then some of the benefit of those experiences get lost.
Yeah, I think that's a great answer. And, for me, I also have Duchenne muscular dystrophy. And, I wasn't diagnosed until I was 5.5. And, thankfully my parents were right on it. But if they could have done that, 3-4 years earlier, who knows, the impact that could have. And I think that's common around all rare disease communities, that diagnostic odyssey, and newborn screening can fix that. So I wanted to follow up with you quickly. What are some challenges to newborn screening? And are there innovative approaches that you're looking at apart from the CK test that can help establish a diagnosis earlier?
Well, the challenges are the infrastructure. And as I said, 50 states in the United States each state will make their own decision. There is in the United States a federal commission under the Secretary of Health that establishes a sort of a guide for rare disorders to add to the newborn screening list that the states may then look to. So the goal for people interested in trying to get Duchenne screening on the books is to get some experience, bring it to that federal committee, get that committee to say, yes, we recommend this disorder to be added to each state's panel. And I think there's room for innovation in how to do the test. The CK test itself is maybe not very expensive, but the sequencing is expensive. And so I think there needs to be, if there could be advancements in high throughput sequencing for newborn screening, it's not unusual for it to take 5 to 10 years for a new disorder to be added to the newborn screening panel on a state. And that's why in the hospital I work in Massachusetts, in Boston, we sort of said, well, we're not going to wait that 5 or 10 years because the therapies are there and they're coming fast. So at least in our hospital, we've just started offering this screening to our pregnant mothers or mothers and fathers. And we decided we'd start in our hospital, see if other hospitals would be interested in doing that, and then gather some experience in our state, and then go to our state newborn screening program and say, look, we can do it. How would you like to take this over and incorporate it into mandated newborn screening for the state? So
going to Dr. Nelson, Dr. Flanagan, the Duchenne clinicians here, once you have that diagnosis at an early age, what's the best way to improve their outcome? And what do you need to learn more to figure out what you can do to improve that outcome?
Well, I'll jump in to say, as Stan mentioned earlier, there's a lot of interest in the idea of treating infants, treating younger children, even to infancy, with steroids. And although there's a limited amount, there is published data on the use of steroids in the youngest population. It's not of the class of data where we can say we clearly understand its effect. But there's sufficient longitudinal data to tell us the earlier we start steroid in patients, the better they do, the more bang we get for the buck. And many of us do use alternate regimens, including weekend dosing in the youngest population, to try to get some of the benefits of steroids without the same side effect profile of daily steroids, particularly in the youngest patients on growth, for example. We are a little off out of the best evidence-based medicine when we do that. We're off into some of the art of medicine, I would say, but there's a consensus among most clinics that see lots of patients in making those types of recommendations. I will say it's challenging because, you know, it wasn't that long ago, it was perhaps 15 years ago, where less than half of the clinics in the country were using steroids for Duchenne. So moving that bar requires increasingly good trials, increasing publications of retrospective data. And I do think that bar is really kind of moving. I agree with Stan. We would use approved drugs like exon skipping in appropriate patients as early as we can. It's quite clear that we all understand we increase the side effect profile of steroids when we treat early like that. So we often have to make best kind of adjustments based on our own clinical experience. And I do think it's important for people to know that really very well-meaning people with lots of experience can differ on the details of what kind of regimens one might use in the youngest patients. But that there's a general agreement that earlier is better under most circumstances. So that would be the two things I think. Well, I'm sorry, let me add one thing. The other thing that early diagnosis allows us is to begin early interventions for those children who have cognitive impairment as part of their syndrome, or other developmental delays associated with dystrophinopathy. So I really think it's imperative to identify patients and get them into the sort of early intervention programs that can maximize their independent function.
Yeah, and I just echo several of Kevin's comments. I think there's a lot of reasons to sort of push for sort of team care very early on in Duchenne for a lot of these reasons. So in the neurocognitive area, you know, certainly pushing for appropriate treatments, referring people to regional center. These are resources within every state that can provide lifelong resources to kids with substantial learning school issues for a subset of them. And those are better to recognize before age 3 than after age 3. And so we really do want to push for some of those things occurring much, much earlier. You know, the other component is, you know, 2/3 of all of our families have no family history of Duchenne. It's either a de novo mutation, often in the mom, who then passes it on unbeknownst to her that she's a carrier, or it's a de novo mutation in the child. So there's no family experience with Duchenne. It's a genetic disease, but it's new in the family. And that's the vast majority of all the people that we see in every clinic. And that's how it's been for a couple decades. And so, you know, it's, as your family knows, as every family, as you head into this, you develop a community, you really have to have a lifelong plan for how you're going to be combating this, how you're going to be living your life, you know, what accommodations you might be looking for, both in how you're living, transporting, all sorts of issues that really play out across the family. And I think the sooner we can be giving people accurate, informed information, accurate guidance of what that looks like, and sort of the team to provide some guidance, the better off we are, not allowing people to sort of have to cobble together their own structures for those aspects, which are, you know, sometimes not medical. And then the others, of course, since they're often new mutations, we need to deal with the genetic consequences of that for risks for other children, other pregnancies, other females, and giving families guidance for, you know, a potential carrier female who's maybe only 3 or 4, who may or may not have some manifesting symptoms, right? So there's a lot of proactive work that I think is actually very important, where, you know, for instance, would be an individual who has a high CK will also have high liver enzymes. And often we see people getting liver function tests leading to liver biopsy without consideration that it's really a muscle disease. And actually, in our own look at UCLA records, that happens about over 10% of our diagnoses trickle into us that way through through basically hepatic evaluations, which are for naught. And Kevin, I suspect that's right. Similar to yours. I don't think it's not an illness of our world here.
Right. We've never checked our numbers, but that experience is consistent with our clinic, certainly.
Yeah. And it happens at UCLA and it happens outside of UCLA. It's not a I got to the Mecca kind of a thing at all. It's just so I think, you know, we can prevent a lot of unnecessary odyssey, but a lot of unnecessary angst in other family members by getting into the heart of the matter and proactively providing that information to family members, particularly parents, but also maybe aunts, uncles, grandma, grandpa. It can go on for quite a while as we sort of figure out what it looks like in each family for how to help answer unresolved sort of questions, if you will. So we can often have the experience that when we diagnose a child, we also diagnose the mom as a carrier, and possibly even another adult who's much older, who's a male, who's affected as well, and just has never been realized to be that, but is at risk then of getting worked up for erroneous diseases. So,
yeah, I think, talking about getting earlier diagnosis, like it allows you to plan ahead with some of these things. Like you mentioned, oh, we're going to need a wheelchair accessible car, when, he's 13 or 14, when he needs a wheelchair to get around. So those are questions. The more we know earlier, the easier we can plan ahead and prevent some of that angst, like you said.
Yeah.
So now I'm going more into, I guess, the genetic component. And, my personal experience is that it's very difficult for me to get into a trial because of my phenotype, mixing my age and my 45 deletion. And I know I'm not the only one who faces the challenges of, trying to get into clinical trials. So Kevin, can you talk a little bit about some of the challenges for genotype, for drugs that are approved for a specific genotype that may leave out a large portion? Kind of what are your thoughts behind that?
Right. So that's we have, as Stan mentioned, 4 currently approved drugs that are genotype specific, and we know of other drugs that didn't reach approval that were, let's say, mutation class specific. And, it is a challenge that these leave the majority of patients without a potential treatment effect for it. There are many challenges with the current crop of those. And I want to be clear here that they've all been approved by the FDA. They've shown some measure of dystrophin expression or phenotypic improvement, although I think it's fair to say those are underwhelming responses in the sense of what we expected when these drugs first came to, or what we saw in animal models, let's say, for comparison. So one challenge is managing expectations with these. Another challenge is getting them. At the same time, we know that most people who use them really are quite aware of the data that suggests over time there is a divergence in the course of disease with patients who receive these from patients who don't. And one challenge is actually in making sure that patients can get access to these. So although there's increasing data, for example, that exon skipping therapies improve particularly lung function and truncal function in patients who are treated with them once they've lost ambulation, I think these still show, I think, a lot of promise, and that's one thing we have to really recognize. The development of that class of therapies is not complete. There are other chemistries coming out. There are other methods of getting antisense oligos into cells that are going to be entering trials that show a significant amount of promise, and in fact have the potential to make more meaningful. They still have the potential to perhaps make more meaningful dystrophin expression than some of the other trials that are not mutation specific. So as an example, I would say that we know very much from looking at sets of Becker patients that the outcomes of exon 45 skipping for certain mutations are really compatible with ambulation long into adulthood, into 40s or 50s. Certain mutation classes that would be the outcome of exon 45 skipping could do that. And we can't say the same for I mentioned microdystrophin, for example. We can't say that for microdystrophin or other things. So we know what the outer limit of what lifelong expression of those versions of the protein should be, which gives us some idea of the function of the protein. And if we can boost expression via approaches like exon skipping, we have the opportunity to approach that outer limit of improvement. In the same way, certain mutations, as one example, single exon duplications, if that gives us a possibility where we can think about exon skipping approaches, really specific things, really specific therapies that are mutation specific that can get us to full-length dystrophin expression. So there's still real promise for mutation-specific therapies. They are not going to be available for all patients, that's true, but they, I think, hold really significant promise.
Okay, great. And I want to get to our last question, just thinking of time here, but this is kind of for all of you. I'll start with Dr. Prad here, but what are the biggest issues that keep you up at night in terms of patient care, those diagnostic tools and finding new treatments? What are you thinking about, in the middle of the night when you can't sleep?
Well, my fears are a little bit removed from patient care. They're more about how I can keep my screening program funded and running, because currently it's kind of it's running on a grant. My fear right now is keeping this going, but we're going to do it as long as we can until everyone else sees the light.
So it's open to Dr. Nelson, Dr. Flanagan, in terms of patient care and new treatments. Go ahead.
Yeah, so I sort of think about things at both ends of the spectrum. Both the earlier diagnoses and rolling out some of these, new, actually rather dramatic, even though not as effective as we'd like so far, therapies. Having a robust clinical trials pipeline that everybody who wants to be on a trial can be on a trial, and then broadening what those trials look like so that we can explore combinations. And that will be absolutely key to how we move forward into this next generation of therapeutics. And then the part that keeps me up quite a bit is, how do we roll many of these things out to those that have, we've managed to care for them for decades, but with substantial loss of muscle functions, or medical abilities to care for patients with Duchenne have been increased dramatically over the course of decades. And can we bring some of these therapies in a meaningful way to them, to repair, modify the microenvironment, replace muscle, those sorts of issues, which are going to be a huge issue for the generation that's already here.
Well, I guess I'm sitting thinking about what keeps me up at night. Maybe there's two things. One is sort of related to mechanisms of some of the newer therapies. We don't know yet about the durability of gene therapies. We're going to learn that question, and that's always in the back of our mind. How are we going to handle retreatment? How will we handle this? And same question with how do we consider treatment with patients who have environmental exposure to AAVs, for example. But those are questions that are being worked on. There's a broader thing that keeps me up quite a bit, which is actually about disparities in access to, not only to the new therapies we're talking about, but just disparities to access for proper care for Duchenne dystrophy. We have to recognize that that those exist, and it's increasingly something that I know a lot of clinics are paying attention to. Why do our clinics not necessarily represent our communities, and who gets access to this care? What are the barriers to these highly these novel, these new therapies? What barriers exist in society, and how can we make sure they're equitably distributed?
Well, I think that's a great way to end our panel here. Thank you so much, Dr. Parra, Dr. Nelson, Dr. Flanagan, for your time. And thank you to our viewers for watching. I hope this was informative, and thank you so much for watching.